drugs? And is there a way to possibly treat this with cell-based therapies? And we'll sort of venture into that in this presentation. So here are my financial disclosures. RPT is a startup company that's developing some of this technology. California Institute of Regenerative Medicine is the funding source for us from the state of California. So in this audience, everybody clearly understands the difference between these two conditions. And we have lots of therapies here, and we're developing complement factors, anti-complement factors here to slow down this condition of atrophic macular degeneration. But the issue really is there a way to replace the RPE in this area? And some of you might say, well, wait a minute, you know, the retina is already damaged. What's the purpose of replacing RPE in this area? Isn't the chordae capillaris damaged in this area? Why are you doing this? So lots of questions, but as I've learned from developing the Argus retinal prosthesis, we had similar sorts of questions. When we looked into the back of patients with RP, the nerve looked pale, and everybody said there aren't any ganglion cells, so what are you doing? And through developing an implant, we learned that yes, there's damage in the retina, but those electronic implants can still use the remaining retinal neurons and still provide useful vision. So similarly, the question really is there enough retina here? And clearly in the end stage, maybe not, but maybe in some stages it is. So I would say the premise is that things are not black and white the way we think of them. And there may very well be some retina here that we could potentially bring back to life or restore if we provided the right RPE cells. But the issue here is that just putting RPE cells is not enough. The Brooks membrane is very damaged, so the RPE doesn't conform or take well in this area. So how do we do this? How do we actually build a very organized layer of RPE? But before I do that, I've told you a lot of things about how the retina may or may not be alive, but where is the data? And here's some very interesting data. So this was autologous RPE cells. So as Dr. Lam just pointed out with Tamer's work, they took the entire retina. But in this case, that wasn't. What they did was did a similar surgery as you just saw, but they just took the RPE and the Brooks membrane. They didn't put the retina, but they did put that autologous sheet here in the macula. And this was what we saw is that visions were very, very poor. But when you did this, visions improved dramatically. And on adaptive optics, you can see the outer segments. So at least there was this, proof of science or proof of clinical evidence that if you did go into area of geographic atrophy and you did replace it with RPE cells, it's a number, about 45% here, that had improvements, but also there was a high rate of complication from PVR and retinal detachments from taking that graft out of the periphery. So this led us to think, well, is there another way to actually not do an autologous RPE transplant, but have something that is safer, but it still accomplish the same goal? So we turned to stem cells, and let me just give you a brief tutorial. Those who are interested in stem cells can obviously read a lot more about it, but in interest of time, there are really only two types, and I'll talk a little bit about this fat cell autologous stem cells. But in the pluripotent types, they can give rise to most cells, and you can either get them from embryonic or blastocyst stage, or you can get it from induced pluripotent stem cells, which means you take, for example, a cell from your skin and reverse program it back to the original stem cell, and then you de-differentiate it into what you want. And Yamanaka won the Nobel Prize for this, for reprogramming iPS cells. So again, pluripotent come in the form of embryonic stem cells and iPS cells. Multipotent can give rise to few types of cells, not all. And adult stem cells do this. There's some of these present in the ciliary body band in our eye, and maybe a source for RPE. But again, the difference between these two, now, you've heard a lot about autologous fat cells. Stem cells derived from autologous fat. You've heard about this in orthopedics. They take some fat, they spin it. So what happened is that there was a clinic, a few clinics actually in Florida that did this, and they injected these cells into the eye, unfortunately leading to blindness bilaterally, and they were charging a lot of money. So the point here being, if you hear about a stem cell study and they're charging patients, it is not the right study. Even if they have clinicaltrials.gov, even if they're on that site, doesn't mean they're FDA approved. These studies were on clinicaltrials.gov, so you would think you'd go on there, you would think, wow, this is an approved study. But it isn't a clinical study at all. They were charging a lot of money, and unfortunately, many people went blind. So be careful about any stem cell studies where people are charging patients, and that is not FDA approved, and clinicaltrials.gov doesn't mean that it's FDA approved. So now, let's go to the stem cell therapies for macular degeneration. Now that you have a little bit of a primer on what stem cells are and what to look out for, and what we need to accomplish. So here you see that obviously the easier thing is to use these suspensions, which I've highlighted in yellow, in red here. And you can see the most advanced is Okada. They've published in Lancet. They have done multiple clinical trials in macular degeneration, also Stargardt's disease. I'll talk a little bit more about this in my presentation. Other suspensions, so these are just injecting cells. And remember what I mentioned about the importance of Bruch's membrane and making sure these cells are oriented properly, but I'll go over that in just a minute. But these are two studies that are going on early stage in China and Israel. The yellow highlights those studies that have already been stopped. So Janssen, which is a subsidiary or big branch of J&J, stopped their trial of using mesenchymal stem cells, and similarly neuroprogenitor cells was being developed by Stem Cell Inc. and was also stopped. Now in terms of the green are now we're shifting here in the black print are those that are trying to create a monolayer, not just injecting a suspension of cells. And again, they come in different formats, but you have the biodegradable scaffold, which means they put it on a sheet that's degradable, is trying to be developed by NIH. It's in the preclinical study, it hasn't entered clinic. Autologous adult RPEs, IPS RPEs, Riken in Japan is really advancing this in phase 1 studies. And then there was a very interesting study where Pfizer was involved in a polyester scaffold that also stopped. So this kind of gives you an idea of the various cell suspension studies here in red, and then the sheet studies here that are ongoing. What we did was, but before I get into this, where is the evidence that suspension is worse or the monolayer is better? Where's the evidence? Just as we talked about, where's the evidence that photoreceptors are present? I showed you from that autologous surgical experience. Here you can see this is a growth factor, PEDF, which is very important to the photoreceptors. And you can see here that when the cells are polarized, this is embryonic stem cell derived RPE, when they're polarized, they do put out a significant amount of PEDF. But when the same cell is not polarized, you can see they don't do well. They don't function well. They don't survive well. And I'll show you this here as an example, some of the studies we had to do, and we'll get to the clinic in just a minute, but this is important to point out that here, when we put it on our substrate, and I'll talk about the sheet, these cells here survive. They have very nice markers. RPE65 is a marker of cells. Here, when we injected the same cells, exact same cells, but as a suspension, you can see that the RPE markers are not there. So some of you in the audience might say, well, wait a minute, what are these pigmented things here? Those are not RPE cells. And interestingly, what they are is macrophages have come in and eaten these RPE cells. So in fact, when you inject them as suspensions, not only do they not line up and provide growth factors, but they also don't survive. Macrophages come in and they eat them up. So how do we get them lined up? So this, took 5+ years to develop, and largely because of we had to develop the world's first synthetic Brooks membrane. So Brooks membrane is obviously, as you know, is not a true membrane. It consists of multiple layers, but in this case, we had to create a scaffold, and we used the substance Parylene C, which has the highest biocompatibility of any biomaterial out there. So USP Class 6 is the classification for this material. We've used it in other implants, but you can see here that this substrate grows these cells very nicely, the basal side being here and the microvilli pointing up. But those in the audience may say, well, and this is a non-erodible sheet, so once you put it in, it doesn't go away. And some of the audience might say, well, why didn't you make it erodible? Why didn't you just have it go away? Well, number one, we don't know when it should go away. I don't know, should it be 6 weeks, 8 weeks, 3 months? And that creates an issue. Secondly, how do things get eroded? Well, macrophages come in and eat the erodible substrate, which sets up an inflammatory response, and we don't want that response in this phase because it could affect the graft. Okay, so now we have this biocompatible sheet with the non-erodible. Cells look good, but you're in the audience are, I'm sure, saying, well, what about the diffusion constant? I mean, these things have to diffuse nutrients back and forth through the choriocapillaris. It just can't be a barrier. Well, we tried this membrane with holes in it, like Swiss cheese. It doesn't work. Cells tend to go right through the holes and don't form monolayers. They don't like it. So we had to use something we've used before, which is through microfabrication, how chips are made currently, integrated chips. What you do is you keep this top surface completely flat, but you go from the back side and etch these thin areas. So these are not full thickness holes. These are etched from the back, and they have to be down to 0.4 microns. That's what allows the diffusion. But then you might say, well, why didn't you just make this whole thing 0.4 microns? Well, 0.4 micron sheet is impossible to surgically hand. It tears, it rips. So we needed to build these thick regions within thin regions. We did a lot of diffusion calculation, and so we ended up with now this synthetic scaffold, which has the same, so this is the molecular exclusion of Brooks membrane, about 200 kilodaltons, and this membrane at 0.5 is at about 291, at 0.4 it's closer to Brooks membrane. Okay, so now we have our cells, which are embryonic stem cell derived cells that go completely into RPE. We're not putting stem cells into the eye. This is a very, very important message. We're deriving them into RPE, getting them to be 99.999% RPE first. Then we're putting them on the synthetic scaffold. Okay, so now we're ready to go, right? No, we have to, well, we're able to put it into a rat pretty well. You can see here the membrane, the beautifully lined RPE with excellent outer segments. Very, this is this part blown up into here, so you can see how well that's oriented. You can also see that these cells actually phagocytose the rod outer segments. You can see them here in the RPE cells. So these cells actually function the way they're supposed to. And we can also record superior colliculus recordings over the area. We can get responses in the rat. Remember, the projections go to superior colliculus, but when we record somewhere else, you don't get the responses. So all this data was put through, but not so fast. You had to develop a surgical instrument to put it in. And how do you do it? If you just put the sheet in, the fluid in the eye during vitrectomy will blow these sheets off. So you have to protect it. And we developed this instrument here, which has a forceps at the tip, but it retracts and folds the membrane in, so protects it in the sheet when we go in, and only when we're underneath the subretinal space, it opens up and delivers the sheet. So you can see here is the membrane next to a penny. We have a handle here in the back where this instrument grabs it and we can move it forward. You can see here that this instrument, this was the early pig studies, but you can see how it grabs it, it folds it like a taco. We're able to separate the retina using hydrodissection, putting fluid in through a small retinotomy, able to put the device in. The other advantage of this device is, of course, you only have to make a small retinotomy to get the device in. If you were to get this sort of device in without folding it, you'd have to make a large cut in the retina, large cut in the sclera. So this allows the whole surgery to be done through small incision through this device. But those who are wondering in the audience are saying, well, wait a minute, if you folded this and unfolded it, don't the cells just fall off? Well, this is some study that Amir Kashani did, who's involved in this, and we'll talk a little bit about Amir's involvement, but we did this pre-implantation and post-implantation, and we had to do this many, many times for the FDA, fold and fold, fold and fold, to make sure that these cells didn't, that they actually survived. So now we have the cells, we have the membrane, we have the tube. So let's see. We did this study now in the pig. So you have to do 2 animal models. You have to do the rat and the pig. You can see here the scaffold with the RPE, the outer segments. Again, well marked for the cell type we want. Very nicely laying. So all this data was submitted. So now 5-6 years have gone by. So we need to get into the clinic, so we've moved forward and we submit a 30,000 page document to the FDA, and we get our first phase 1/2A approval to do the study. So now we're here, we have 20 subjects were given. They're in 2 cohorts, 10 for each. They have geographic atrophy, so that's the disease we're going for here. And the first 10 have to have very bad 2400 or worse vision. The second cohort can be 2100 initially, now 2080. So here's the vision inclusion criteria. And so we get started here, and this is the surgery. Amir Kashani here is the PI. We've already done the vitrectomy, field of hyaloid. So a lot of people worry about hydrodissecting the atrophic retina. You have to use a special cannula there with hydrodissecting it. It does take some time. Here's that special forceps. Now we're in the eye. We've hydrodissected it. And you can see we have a small retinotomy here. And you're able to unfold this. And this is Amir Kashani unfolding this underneath the retina here. You can see the membrane now. It's very clear to see. Through a small retinotomy, you're able to get this in. Once the membrane is in there, we're able to use the handle. Remember, you can grasp at the handle. Some of the cells will come off there, but that's out in the periphery, outside the area of geographic atrophy. We make sure it's laying flat there, and then we put perfluorocarbon in to evacuate the fluid that's there. You don't want to aspirate here at the retinotomy because you might take those cells out, so it's better to use PFO to flatten it out. There's the membrane you can see, and we move forward. So here, for example, is one of our subjects. You can see bilateral geographic atrophy, long-standing, and you can see here very nicely this membrane put into this eye. Here's the pre-op again and the post. And again, through a very tiny retinotomy out here, you're able to get this large membrane in which covers the entire area of geographic atrophy. So this is the exciting and last few slides. So what did this thing do? After all, we went through this and spent all these years. And what you see here is post-op day 2. You can see this membrane that we talked about, this Paraline membrane. We engineered it such that it has a 1.5 refractive index. So what does that mean? The tissue has 1.3, so you can clearly tell where your membrane is. You're not going to get confused. That's one of the advantages of it. You can see these cells here pretty clearly sitting on top at post-op month 2. But what's very, very exciting and has never been shown before is the presence of external limiting membrane. You can see it out here. It wasn't there post-op day 2. It wasn't there preoperatively, but you're starting to see it, and you see it out 6 months too. So, and the cells are there. It's important to show 6 months because we stop the immunosuppression. These patients are, this is not, obviously this is not matched with them, so they do get tacrolimus for about a 6 to 8 week period. And this is after tacrolimus is stopped, and you can clearly see that the cells are still there. So external limiting membrane, why are we so excited about that? So here, external limiting membrane really only is a delineation between the nucleus of the photoreceptor and the inner segments and outer segments. And this brings us to that whole area of thought that I started, and why these patients improve or potentially improve is because if there's no RPE, well, outer segments are gone, and maybe, but what happens is the nuclei may be very much there. This whole concept of dormant nuclei. Why do we think that if the RPE is gone, the nucleus goes? And in fact, this is a very interesting area that these nuclei may be there. And so once we put the RPE here, they start to grow their inner and outer segments. So obviously, this is a hypothesis, but you don't get external limiting membrane formation unless you have these. Now, what we are doing is, in addition to the structural information, what's very, very important is, where's the functional information? Well, you'll have to wait because we're getting our publication, hopefully, right, Amir? We're in a very high-profile journal, which is reviewing, and we're in final reviews, where we do have functional information, and there is functional improvement. But you'll have to wait and read that journal article because it is embargoed at this point. We can't talk about it. So I can take you this far, show you the structural improvement, explain what we see, but you'll have to wait for functional results. And here, I'll show you a cell suspension in comparison. Okada, remember we talked about that. So these folks are injecting embryonic stem cell derived RPE into the subretinal space of geographic atrophy. What is the difference? You can see here, here's suspension from the study, no RPE, no external limiting membrane. Here's when you put it down and on the sheet, you get the RPE and external limiting membrane. Very different construct in the same disease when you do put a monolayer that's organized and polarized the way you want to. So I've walked you through a lot of new information, hopefully haven't confused you. There are different types of trials that are going on, different mechanisms of action. A suspension cell that doesn't form a graft may be working through a different growth factor mechanism versus a graft, different modes of delivery, suspension versus sheets. I need to wait the results from the trials, and I'd like to really thank our clinical trial site. Mark Bearcat's here. We work with Praveen, as well as I mentioned, Amir Kashani's the PI here. Faraz, Rahal, Bob Avery, Clement, and Sanford Chen have all been very helpful in recruiting for our trial. And lastly, on the USC side, in addition to myself, David Hinton, Amir already mentioned, Debbie Mitra and Bijou Thomas did a lot of the animal work. UC Santa Barbara does a lot of the stem cell differentiation work with Dennis and Link. At Caltech, we developed the membrane with Yu Chong Tai. And City of Hope is where they make the product, so you need someplace that can make it to enter clinical trials. CERM is our funding source. Jane Lepkowski is at RPT, Regenerative Patch Technologies, I already mentioned the clinical sites. The medical monitor and DSM are Mon Nasser and Suber Wong. So thank you for your attention.